Thanks everybody for coming. My name is Fritz Klassner. I am the Natural Resource Program Manager with the Office of Maunakea Management, and this is the monthly Maunakea Speaker Series. Tonight's a little special night. They're all special, but tonight's an extra special night because of, we have a suite of speakers. The Hilo High Science Search class or Science Fair program with Miss Mya Lee in the back here. She recruits her students to talk about their science fair projects. There's 6 students tonight presenting, and I think 4 of them are going to a science fair this weekend in Honolulu to present. This is their dry run before their science fair this weekend. And the other 2, this is a last chance to give their science fair talks before graduating and moving on. So first up tonight is Jared Goodwin. Jared is a senior at Hilo High, as everybody's at Hilo High, and he's going to be talking about his windshield wiper model for arsenic after the 1960 Chilean tsunami.
Okay, so hello, my name is Jared Goodwin. I'm a senior at Hilo High School, and my project focuses on mapping the distribution of arsenic contaminants in and around Wailoa State Park. Quick introduction about myself. So my family's been involved with tsunami disasters in Hilo, Hawaii for over 3 generations, most notably my grandmother, who actually had to survive the 1946 Aleutian Island tsunami, which eventually destroyed her home, and they were actually sent running into a nearby temple. And so tsunamis have had a very big effect on my life, and basically it has influenced for the past 4 years my science fair research. And a little bit more about myself, I've been involved in soil-based science fair projects for over 3 years. It actually began when I questioned if different types of land use would actually affect different concentrations of heavy metals. And so this project that I'm presenting here is year 3 of a ongoing 4th-year continuation research project. And in the first year of research, I wanted to answer a yes or no question, which was, did tsunamis possess the ability to distribute heavy metal contaminants such as arsenic and lead? And so in the first year, it prompted me to conduct a large land survey of the land surrounding the 1960 Chilean tsunami inundation zone. And data from that year found that locations primarily along the inundation border further outside contain much greater concentrations of both lead and arsenic for all locations. And so that actually prompted me to form what is known as the windshield wiper model, which is a model I formed during the first year of research, which essentially aims at predicting and describing the movements of these heavy metals through the forest of tsunamis. And moving into my second year of research, which was in my sophomore year, I decided I wanted to take this model and further test it and its principles in a location which is Wailoa State Park, which also contains a body of water known as Waikea Pond. And the reason for this is its history, which we will get into a bit. And the third year of research, or the project that I'm presenting now, actually I wanted to create sort of a spatial map or a hotspot map of these contaminants to see if I could see some sort of gradient of concentrations that agreed with the wave patterns, to see if I could possibly validate this model further. And so as for the purpose of this, this presenting project is I wanted to essentially survey the land surrounding Waikea Pond, which is Wailoa State Park, and I wanted to further validate my windshield wiper model. And in this, my hypothesis was that arsenic concentrations in a hotspot map would be able to resemble sort of wave pattern gradients, and also this is that we knew where the source of arsenic was being put out into the pond from a production plant. And my hypothesis aimed at saying that concentrations would be greatest nearest to the source of arsenic due to the possible distribution by tsunamis. And so a little bit of background on my project. So there is something known as NATEC disasters, and these are known as natural disasters, which can trigger technological mishaps that can release unwanted pollutants into the environment. This can be both destructive to humans and ecosystem factors. And so an example would be the 2011 Tohoku earthquake and tsunami, which actually took out 3 Fukushima Daiichi reactors. It killed the cooling reactors and thus resulted with the cores melting and released untold amounts of radiation to the atmosphere and the surrounding soil. And so it is no different when it comes to the talk of the 1960 Chilean tsunami, which had the destruction and caused, more than $300 million of damage to Hilo town at the time, and possibly the distribution of these heavy metals. And so we also know that arsenic can actually mobilize in both groundwater and river water sediments. As issued here in the Hokusetsu area of Japan, we actually can see that these arsenic contaminants were transferred from riverbeds to different locations within parts of the river. And so the question usually is, what is a tsunami? So tsunamis are, it also translates over from Japanese to harbor waves. And what these are is these waves are giant sea waves that are generated from seismic activity. They can usually range anywhere from about 8 to 13 waves. Usually the third wave is the strongest wave. And they also, again, they can be classified as NATEC disasters, and they have the ability to disperse these hazardous contaminants. And so getting into really why I chose Wailoa State Park as an area to test my model with a body of water. So as pictured here is what is known as the Koenig production plant. So this production plant was built in the late 1800s and was operating until 1963. This production plant was headed under Flint Coke Company after they bought out it from Hawaii sugarcane in 1948. And it was estimated from 1932 to 1963 in a 1990 NOAA study, an estimated 558 tons of arsenic compounds were being released into the pond. And these arsenic compounds were used as an anti-termite treatment for this dry board material known as Koenig. So Koenig was a dry board material that was commonly used in construction. And this is material that was sourced from sugarcane stalks that were extracted of their juices. The boards themselves were not of any harm to humans. However, the termite treatment that they underwent with the arsenic compounds was. And so along with the contaminants being poured into the pond directly, a lot of the homes were essentially laced with these contaminants. You would say primarily from the 1950s to 60s, you can find any homes built within that time. A lot of the ceiling and the wall siding, the panels, contain very high concentrations of arsenic because of the Koenig dry board. And so the whole thing is, what is arsenic? So basically arsenic is a naturally occurring element. It occurs in the soil. Particularly, levels are much higher in Hawaii, and the reason is that our composition calls for a lot of iron in the soil, and arsenic likes to bind to iron, so total arsenic levels can be very high here. But at times it's not too alarming, as there are different types of arsenic. You have ones that are actually harmful to us and ones that are naturally occurring in their organic form. And so the ones that we're analyzing at Waikea Pond are actually inorganic compounds, meaning they're compounds that are actually manipulated by humans. And so in fact, the health effects that can be retained from actually ingesting arsenic, whether it be through soil or contaminated water. So short-term effects, usually people exhibit anything of vomiting, diarrhea, skin lesions is a really big one. And then, death in extreme cases, which is extreme if exposure is super high for a short amount of time. Long-term effects can include anything from skin cancer and mostly organ cancer, so such as the lung, bladder, and kidney. And in rare cases, also death. And so getting more into my model. So my model has 3 principles, the windshield wiper model. And quick overview of why it's actually called the windshield wiper model is we would think of the land that's being inundated by the tsunami as a windshield, and we would think as the tsunami wave being a windshield wiper. Tsunami waves have an inundating and a receding period, and we would think of those pre-existing heavy metal concentrations as water droplets atop a windshield surface, and with an inundating and receding period, similar to a windshield wiper movement, you would have an inundating and receding motion. And so the 3 principles are actually, each principle is derived from each year of research and the data that's presented to make any conclusions. So the first year was actually the windshield wiper model affecting dry land area. So this is the large land survey, and that found that locations along the border further outside contain greater concentrations. The second principle, which was the first year of testing at Waikea Pond and Wailoa State Park, was that heavy metal concentrations were actually greater from the embankments of Wailoa State Park pond, Waikea Pond. And this is because supposedly those concentrations within the pond were distributed to different locations over the embankments. But tsunami waves are much weaker receding than they are inundating. And so it's possible that these concentrations of arsenic could have been entrapped in geological land features such as topography, which is a difference in altitude, and also, you know, the barrier being the embankment itself. And so the third principle, which is also up for testing in this project, was that concentration source from a point origin would actually be greatest nearest to the source of heavy metals. And so this is a little bit of a illustration of the windshield wiper model for better view. So we would think, again, as the wave being the windshield wiper, and we would think as all those arsenic concentrations within here as the water droplets. And so these red bubbles, as you can see right here, actually represent, they are derived from a USGS topographic map. And those numbers actually represent the land in feet above sea level. So as we can see, we near the soccer fields that are located by Hilo Bay Front, that land is around 4 feet above sea level, so fairly low. However, we start to see as the land starts to increase to from 10 to 17 feet above sea level. And also, we have a very high point by Suisan, which is 13 feet above sea level at the mouth of the river. And as we see, it begins to decline to about 10 to 12. And so moving on to the methodology. So there are a total of 80 locations spanning Wailoa State Park soil surrounding Waikea Pond. And there are a total of 240 soil samples. And so these samples were plotted and mapped using GIS technology. As such here, it was actually a digital application on a phone. And so I was able to get longitude and latitude readings in UTM. And so with the soil collection, so the reason why there were a total of 240 soil samples from 80 locations is that at each location, there were 3 layers of soil that were tested, layers A, B, and C, with A being the top layer and C being the bottom layer. And the reason for this is that arsenic is known to sink in soil at a rate of about 1 to 2 inches per every 10 years. And since the almost 60 years it's been since the 1960 tsunami, it's plausible that these concentrations could have sunk to a maximum depth of about 1 foot. And we also can see sedimentation taking place here. You see a visible band of sand that was actually similar in composition to sand that's at Hilo Bay Front. And then we have a lot of the soil that's surrounding it here. And so these samples, of course, were derived using latex gloves with proper safety precautions. And then part 3 of this was prepping the sample for what is known as an XRF machine. So this is basically preparation for testing. So each individual sample of the 240 set, each of them were baked for about 3 to 4 hours using a home conventional oven for about at around 300 to 400 degrees Fahrenheit, depending on the strength of the oven. And then they were transferred into these little containers, which contain about 5 grams of soil. And we actually have a film of micron, which is actually a transparent film that's placed over the soil sample in a special cup. And as seen here, so this is known as a XRF machine, which stands for an energy dispersive X-ray fluorescence technology. And so this actually gives a non-destructive elemental composition of soil samples within a 10 to 20 minute runtime. And so if we look at the wheel here, and this is actually a visual of the wheel inside the machine prior to X-rays. So the way XRF technology works is it uses 2 forms of X-rays, as we can see here. So one source of X-ray comes from the machine itself, and it begins an excitation process with the sample. And after about 10 to 20 minutes of runtime, we get what is known as secondary forms of X-rays, also known as X-ray fluorescence. And what the machine measures is these ejected electrons as a result of this excitation. And this is how we were able to get readings of arsenic, lead, mercury, and zinc, and so on in readings of parts per million. And so we move on to the data. And so this is a XY scatter plot. So this, there are 3 different graphs, all for each layer of sedimentation. And so the X plane represents the distance from the proposed source of arsenic, which was near the peninsula of the southern peninsula of Waikea Pond, which is now Waikea Villas. And on the y-axis, we have the arsenic concentrations in parts per million. And this is also to note that the safe level of arsenic concentrations by the EPA is 20 parts per million. However, the Hawaii Department of Health designates that due to our elevated concentrations of arsenic, 24 parts per million is considered a safe level of arsenic. And so fairly in distance, we see that within a 400-meter range of a total of 800-meter span, we actually see that there's a borderline cluster of arsenic concentrations within the elevated range, which is around 51 to 150 parts per million. And for the first layer of soil, the greatest outlier was actually nearest to the source, and that was around 997 parts per million. And so as we increase with each layer, we actually see that those outliers and also the borderline cluster of these concentrations gradually increase to around 1200 parts per million. And finally, in layer C, we have 2 concentrations that are actually greatest between 1,728 parts per million and 1,729, which is well over 400 times the safe level of arsenic. And as far as the map, so this is the windshield wiper model's visualization. And this is the gradient of arsenic concentrations that were tested. So blue would represent any concentration that fell into the safe level. And this map was created using average concentrations, so over the 3 layers, from layers A, B, and C. However, unfortunately, we don't have any blue bubbles here. But we do have concentrations that range from within the moderate level, which is anywhere from 21 to 50 parts per million. And also the red, which represents the extreme, which is anything greater than 501 parts per million. And so we actually noticed that there is a gradient located along here. So the waves of the 1960 Chilean tsunami came in at a southeastern direction, sweeping along the east coast of the Big Island, thus into Hilo Bay. And so we actually can see that these concentrations heavily resemble a wave gradient that would actually inundate Wailoa State Park. Any of these concentrations were thought to have been brought out over the land. And to scale, a location located here is about 40 feet from the embankment of Waikea Pond. And it's also noted that there was another point of entry from the arsenic. So there is a pipe that ran adjacent from the Koenig production plant and out here near the river mouth. And that is another source of arsenic where they said they were actually dumping the arsenic directly into Waikea Pond. And so it's plausible any of those concentrations within the pond could have been distributed from within the pond to locations outside the pond to dryland soil. And so essentially when we look at those depth layer scatter plots for each layer, we saw that there was obviously a baseline cluster of arsenic concentrations within that yellow or the elevated range up to 150 parts per million. And then we also saw that, arsenic concentrations gradually increased with depth. So we saw, that sinking rate. And moving on to the hotspot of Wailoa or Waikea Pond, we saw that, the greatest arsenic concentrations were actually in locations farthest from the inundation zone. But we also see that there is a possible effect of topography playing. So locations here were actually located 10 feet above sea level, as previously noted in the topography map. And locations here were about 4 feet above sea level. However, there is a possibility that these concentrations could have been a result of entrapment as well, as the land surrounding it is around 13 feet above sea level. And so while that 3 feet difference in topography may not seem as big as it is, you have to think it's a gradual increase, but it is still an increase. So those concentrations in the stronger inundating period of the wave could have been entrapped through the weaker inundating period or receding period. And so with the windshield wiper model, the first principle is ultimately refuted whenever we are dealing with a tsunami-affected body of water that's been polluted. Reason being that, there are concentrations that were near the inundation zone, but ultimately weren't as great as concentrations that were within. as any of those concentrations within the pond were actually derived to locations outside. Principle number 2 was ultimately supported because locations farthest from the embankments were greatest. And when it came to the third principle, it was partially supported because within that 400 meter, there were still a relative number of concentrations, you know, nearing up to 800 meters from the point source of arsenic that were greatest. And so coming to conclusions here, so there were no average arsenic concentrations of any sample that met the EPA safe level standard of 20 parts per million, or even the Hawaii Department of Health's guideline, which is 24 parts per million for Hawaii. And we also see that topography did have some sort of effect with arsenic contamination, and also that the model was supported, but there are revisions that need to be made to the principle, and that there are, you know, we ultimately see that there is a gradient and a visible distribution of these contaminants by a tsunami. And so future steps is actually what I am ongoing here. So I'm currently in my 4th year of research. And right now, you know, the whole thing is to increase overall sample collection. The more data, the better. And also I'm currently working with the Hawaii Department of Health we're retesting samples that were taken in 2014 with no current tsunami event that could have affected these results. And so we're actually testing concentration that came from within Waiakea Pond. And so my hopes is that we're able to create sort of a hotspot gradient, but also a spatial map of these concentrations within the pond as well as outside the pond. Comparing these results within the pond and outside the pond, we'd be able to see if the tsunami had actually had a direct and sure effect on the distribution of these concentrations. And lastly, this has come to propose and introduce forms of environmental remediation. So as Mr. Klasner mentioned, I was humbly able to be inside of a documentary known as Inventing Tomorrow. And so it runs alongside myself and 5 other students from around the world dealing with environmental issues facing their own backyard. And the topic of Wailoa State Park was one of the headliners in here. And so what I am seeking now is more of a public outreach and getting this data out to different people, possibly publishing the study with my mentor in the next couple of years, to at the least get signs up in the park to warn park goers of the possible danger. And applications to the real world. So when it comes to how is this project applicable to other places, is that my hopes is that when I am able to finalize the principles of my model, I hope to apply it to different coastal regions that have been affected or are susceptible to tsunami disasters, such as Sunda in Japan, Indonesia, we just faced the Indian Ocean tsunami. And there, if the model is accepted, we can apply it there to see exactly where these arsenic cleanups are most needed, whether it be for any type of other heavy metal. Thank you. And I'd be happy to answer any questions.
You run across as far as remediation?
So I have talked, so last year in the science fair, or actually 2 years ago in my district science fair, I talked to a biologist and he actually said they're trying to implement some sort of phytoremediation. So this is the application of actually planting crops to actively uptake mostly arsenic 3 and 5. The only trouble with this is that a lot of the plants that are very useful in arsenic uptake are usually edible crops such as rice and tomato. One of the other ones they're testing is tea leaf. And, you know, it begins that search for, you know, what is the most viable option. There has also been other options such as adding phosphate to the soil. But as we know, if phosphate enters the water in excessive amounts, we can get eutrophication and an overgrowth of algae. And the other one, which is the most invasive but the most sure, is soil removal. So this is actually taking place on, there's a corner lot that's been for sale for a while near Waiakea Villas. And the thing is, they've been, from what I hear, they've been getting readings at least 2 times over my greatest outliers in this research project. So they're actually undergoing soil removal, which is a removal of 1 foot minimum of soil. That soil is actually taken to a landfill, a designated EPA landfill, and new fresh soil is brought in. And other than that, there's a possibility of bacteria that can actually absorb the arsenic, as arsenic doesn't deteriorate over time. Thank you.
Next up is Shui Wen. Shui is a 10th grader, again at Hilo High. Shui is going to talk about ohia, or Metrosideros polymorpha, and rust.
Hi, my name is Shui. I'm a sophomore at Hilo High School. Today I'll be presenting to you my research on Metrosideros polymorpha, different wood tissue types, and their susceptibility to the fungus Ceratocystis zucoohia. Metrosideros polymorpha, more commonly referred to as ohia, is the most abundant native tree in the state. It is endemic to the main Hawaiian Islands, and due to its abundance and extreme environmental variability, it's considered a keystone species. It provides a habitat for Hawaii's many native birds and arthropods. The tree itself is also very culturally significant. Many Hawaiian heiaus are made of ohia wood, and the plant itself is considered sacred to Pele. Rapid ohia death is a disease that's killed off over hundreds of thousands of ohia trees on the Big Island, and it was recently discovered on Kauai as well. So there are two fungi responsible for this disease, Ceratocystis huliohia and Ceratocystis leucoohia, of which the latter is more aggressive, which is why I chose to incorporate Ceratocystis leucoohia into my experiment so that I could see the worst possible case scenario that this disease could become into. So the disease enters the plant through a wound in the plant, and it grows in the xylem sapwood area until it blocks off all the waterways, basically drying the plant out. Ohia propagation is the replanting of ohia, and the most common way to do this is by collecting cuttings off of already existing ohia plants. Unintentionally, ohia propagators are leaving wounds on the plant exposed to the fungus. These wounds are not thought to be able to be infected by the fungus, but I wanted to see if these semi-hardwood tissue wounds on ohia plants were able to be infected, and if they were, how long it would take for symptoms to start appearing on the inoculated plant. I hypothesized that these semi-hardwood tissue wounds would not be able to get infected by the fungus. This is because semi-hardwood tissue has a very small diameter, and that leads to a small amount of xylem vessels in the wood, which is where the fungus grows. So I figured that the fungus would not be able to spread rapidly enough, and that the plant could basically isolate it. For my first experiment, there were 16 plants in total, with 4 experimental plants processed each week. To inoculate the experimental group, I had a filter paper disc with the inoculum growing on it. I placed the disc on the wound, which was on semi-hardwood tissue, and I wrapped it in parafoam. The steps for inoculating the positive controls, I used the same filter paper disc with the inoculum of the fungus, but this time I inserted it into a cut in the main stem of the plant and then wrapped that in parafoam as well. For the negative controls, I repeated the steps that I used to inoculate the experimental group, except this time the filter paper disc did not have any fungus on it. It was simply soaked in sterile water. So at the end, the experiment lasted for 3 weeks with 4 to 6 plants processed each week. The steps for processing the plants were I first unwrapped the parafilm and then scraped off the bark just enough to reveal the staining caused by the fungus. And then I measured the staining length using calipers. A method we used to confirm the presence of the fungus was called carrot baiting. We simply sandwiched the infected wood between 2 carrot discs. Carrots are a surprisingly good host of the fungus. So we left that carrot sandwich aside, and then a week later we looked at the disc through a microscope and searched for the presence of parathecia, which is sexually fruiting bodies of the fungus. You can see in the top right photo the parathecia, which is the sexual fruiting bodies of the fungus, which confirmed that the staining was indeed caused by the fungus and not any other factor. We also did the carrot baiting test for the negative controls as well to make sure that there was no fungus present. The results show that the semi-hardwood tissue was indeed susceptible to the fungus Ceratocystis leucoohia. At the end of the 3-week period, there was an average staining length of 80 millimeters. And in the second graph, you can see that there was already staining apparent in the first week. after inoculation. So this answers your second question, which was how long it took for symptoms to appear on the plant. It takes less than a week for the fungus to settle into the host. These are just some symptoms of the fungus. You can see the wilting and the yellowing of the leaves, and in the middle picture, if you look closely, you can see the darkening of the dark spots in the main stem. Next, I wanted to compare the susceptibilities of the different wood tissue types within the ohia plant to the fungus Ceratocystis ohia. So the three wood tissue types were succulent tissue, semi-hardwood, and hardwood tissue. First, I wanted to see if succulent tissue was able to be infected by the fungus. I hypothesized that wounds in the succulent tissue type of ohia would not be able to get infected by the fungus. Again, my reasoning was the same for my last hypothesis. Since the diameter is so tiny, there would be a small amount of xylem vessels, and the fungus wouldn't be able to spread rapidly enough to take over the plant, and the plant could isolate it. When comparing the three different wood tissue types and their susceptibilities to the fungus, I hypothesized that the average staining length of succulent tissue would be the lowest, and that hardwood tissue would have a greater staining length than that of semi-hardwood tissue. So my methods for inoculating the plant for this experiment were the same as my previous experiment, except this time there were 10 plants in total, and I made 3 wounds on each plant. Each wound was on a separate branch to make sure that staining between the 2 wounds wouldn't overlap. And each wound was also on a different wood tissue type. So we had succulent wood tissue, hardwood, which was already ligmented, and semi-hardwood tissue, which was going through the process of ligmentation. My methods for inoculating the positive control were exactly the same as the first experiment. Again, for the negative control, I repeated the steps as I did for inoculating the experimental group, except this time the filter paper discs were soaked in sterile water and it had no fungus on it. At the end of the 2-week period, the duration of the experiment was determined by the results of the last experiment. After 2 weeks, we saw enough staining to really measure it, and not too much where it would overlap between different wounds of the same plant. We found out that succulent wood tissue was indeed able to be infected by the fungus. And when comparing the 3, we saw that succulent wood tissue had the lowest average staining length, and that hardwood tissue had higher than that of semi-hardwood tissue. In conclusion, the first experiment disproved my hypothesis. Semi-hardwood tissue was able to be infected by the fungus, so we can assume that the semi-hardwood was viable for fungal colonization, and it took less than a week for the fungus to settle into the host. The second experiment showed that the fungus was able to grow in succulent wood tissue. It had an average staining length of around 8 millimeters after 2 weeks. When comparing the different wood tissue types and their average staining lengths, we saw that succulent wood tissue had the lowest average staining, and the hardwood had greater average staining length than that of the semi-hardwood. So what the data suggests is for ohia propagators, when collecting cuttings, to perform preventative measures, and this can be anywhere from wrapping the wound in parafilm to maybe spraying it with a 80% ethanol spray, to perform preventative measures after collecting cuttings, and this is to reduce chances of infection to the fungus. And since the second experiment showed that succulent wood tissue was also viable for fungal colonization, ohia practitioners, culture practitioners, should be wary of this before proceeding to injure the plant by harvesting flowers, etc. Since ohia is so deeply rooted in the Hawaiian culture, I think it'd be demanding to just prohibit the use of ohia trees or harvesting ohia flowers. So we just suggest that ohia cultural practitioners acknowledge this before proceeding to injure the plant. Thank you. If you have any questions, I'd be happy to answer them.
Yes, I have one, Shui. So you mentioned the potential of injuring the plant when you're harvesting. Were you able to see how, if you are moving a piece of harvested material around, does that increase the potential of introducing the fungus to another location?
So the fungus stays present and alive in the wood up to a year. So it does it does spread the fungus around if you transport ohia wood. So the Department of Land and Natural Resources advises citizens to not transport ohia wood or ohia trees, especially into the island, since we want to contain the fungus. Oh, so the negative controls were caused, the staining on the negative controls were caused by other factors. The plant was trying to kill itself off as a self-defense mechanism, pretty much, and it wasn't caused by the fungus. We saw this by performing the carrot baiting test on it. We saw no presence of parathecia, so that's how we know there was no fungus and that it wasn't caused by the fungus.
Thank you very much. Elizabeth Atkinson is a senior at Hilo High, and she is one of 2 students who worked on rat lungworms.
My name's Elizabeth. I'm a senior at Hilo High School, and my project focused on the development of a recombinase polymerase amplification assay for detecting Angiostrongylus cantonensis and comparing it to a quantitative PCR. So Angiostrongylus cantonensis is more commonly known as rat lungworm. It's a parasitic nematode that causes eosinophilic meningitis or damage to the central nervous system in humans. It grows and reproduces in rats, which are the definitive host. Rats become infected by eating infected mollusks, such as slugs. The nematode enters the rat's bloodstream and molts into L5 larvae in the rat's brain, and then lays eggs in the pulmonary arteries of the rat. Mollusks become infected by eating the larvae that exit the rat through their feces, and humans become infected by coming in contact with the infected mollusk, such as consuming the mollusk or just coming in contact with it. And in humans, the nematode takes a similar path to the rats, where it travels to the brain and central nervous system, and then molts to L5 larvae, which is what is thought to be the last stage in humans. So the purpose of my project was to create an isothermal diagnostic test that could be compared to quantitative PCR. This isothermal test, I decided to use recombinase polymerase amplification, and this test is fast and portable. I believe that this assay would be comparable to quantitative PCR because the structure of the primers and probes used are similar. Quantitative PCR is a DNA amplification method that is used to diagnose Angiostrongylus cantonensis in slug tissue as well as in humans. It uses a thermocycler, which cycles through 3 different temperatures to initiate the reaction. A single cycle or a single reaction takes about 1.5 hours, plus an additional 30 minutes to an hour of setup time. In the reaction, the heat is used to denature the DNA strands to allow the primers and probe to anneal, and then to amplify the DNA segment. RPA is also a DNA amplification method, but is isothermal, meaning that it occurs at a constant temperature. The reaction takes about 15 minutes, and with about a half an hour of setup time. It uses magnesium acetate to initiate the reaction, which then allows the different proteins to separate the DNA strand and amplify the DNA segment. So for my project, I first started by collecting slugs from around the east side of the island. I performed a tail snip and extracted the DNA in these slugs, and then ran them with PCR. I ended up finding that all of these slugs were positive for rat lungworm. I then had to design my RPA assay. I used DNA sequence analysis software, as well as an RPA instruction manual that gave suggestions on the primer and probe composition and length. I used the DNA sequence analysis software to visualize the ITS1 region of the DNA, of the rat lungworm DNA, and I could go through the sequence and try out different primer and probe composition nucleotide sequences to see how they would most likely fold when they're created. I ended up finding one probe that didn't have any hairpins, meaning that it didn't fold over on itself or wasn't likely to fold over on itself. And I also chose 5 forward primers and 5 reverse primers that I would test with the probe. I then tested all the possible combinations for the 5 forward and 5 reverse primers with the probe, and chose the primer pair that had the highest maximum slope. This means that it had the most amplification in the shortest amount of time. The table shows a comparison between the qPCR and RPA assays and the nucleotide sequences of the probe and primers. I then wanted to test the accuracy of my RPA assay, so I ended up choosing 44 samples, 10 of which were samples that I had collected. I then tested these 44 samples with both qPCR and RPA. I also wanted to test the sensitivity of my RPA assay. So to do this, I chose one of the 44 samples that came up positive in all replicates for qPCR and RPA that I knew that had a high enough DNA concentration to where I could make 8 tenfold dilutions, ranging from 100 nanograms to 10 to the negative 5th nanograms of DNA per microliter of template. I then tested these 8 serial dilutions with both qPCR and RPA in 3 replicates. The image on the left shows a completed RPA reaction that is under a UV light, and you can see the fluorescence in the top row, which shows that the samples in that top row were positive compared to the bottom row, which did not have any fluorescence. So for the results for my primer performance, I compared the maximum slope and the time that the reactions took to cross the cycle threshold for each of the primer pairs. The circled, the point that has a red circle on it is the pair that I tested that had the highest maximum slope and crossed the cycle threshold in one of the shortest amounts of time. I decided to focus more on the maximum and having a higher maximum slope versus crossing the threshold in a short amount of time, because I felt that having a higher maximum slope meant that it was annealing to the DNA template more efficiently. These are the results for my comparison of accuracy for the 44 samples. The samples that are highlighted with green matched between qPCR and RPA, and I ended up having 23 samples that did match. The red samples are samples that did not match, meaning for either qPCR or RPA, one of them was positive, while the other was negative. The blue, and I ended up having 3 samples that did not match. The samples that are highlighted with blue, I labeled as equivocal, meaning that for not all 3 replicates, they came up positive. For qPCR or for RPA, it didn't meet one of the 2 criteria that I chose to determine if it was positive. The 2 criteria that I chose were the cycle threshold and the maximum slope, both of which I calculated using the controls that were run with the reaction. Most of these samples did have a very low DNA concentration, which does show that qPCR was able to detect the DNA in at least some of the replicates that I performed, while RPA was not able to, which does show that RPA is possibly not as, the RPA assay is possibly not as sensitive. For the comparison of sensitivity, for the 8 tenfold dilutions, I found that qPCR was able to detect down to the lowest dilution that I tried, although not consistently, while RPA was only able to detect down to 10 to the negative 2nd consistently. It can be seen that the sensitivity does decrease for both dilutions as the DNA concentration decreases. So from the comparison of accuracy and sensitivity, it can be seen that the RPA assay does work, although it might not be as sensitive as qPCR. I do believe that it is comparable and that it did provide fairly consistent results. With further development, I do believe the RPA assay could become more sensitive and could possibly replace qPCR in some aspects of diagnostics. The minimal amount of equipment needed for RPA means that it can be performed in the field or in a doctor's office. The shorter reaction time also means that the reactions can be done faster, which means that it's more efficient for if it were to be done in doctor's office or in labs, that it'd be more efficient. I also believe that it could be adapted to human diagnostics, which would allow doctors to be able to test their patients a lot more quickly, because right now qPCR is what is used. The main differences between qPCR and RPA is a reaction time, as well as the amount of equipment that's needed. RPA uses an inexpensive fluorometer, while qPCR uses the more expensive thermocycler, which means that RPA would be a easier option for labs to be able to perform these tests. To increase sensitivity, I think that I could try different primers and probes in different locations, as well as possibly adding more template into the RPA reaction. I did try to match the amount of template added to qPCR and RPA, but adding more template would possibly increase the sensitivity. Currently, I'm working on repeating the serial dilution so that they are 2-fold instead of 10-fold, so I can pinpoint exactly where at the exact cutoff point for RPA. I'm also working on adapting my RPA assay to a lateral flow assay, which is even faster and more inexpensive than RPA because it does not need a fluorometer. The image shows 2 lateral flow strips that I've recently tested. The one on the control strip has only 1 line showing that it is negative, while the one on the left has 2 lines. Thank you.
Any questions for Busy?
Yeah, I know that they are working on blood-based diagnostic testing. Oh, sorry, I know that they are working on blood-based diagnostic testing, testing the blood instead of the cerebral spinal fluid, but I'm not sure how that is progressing right now. The actual strips are available. The only difference was that they used specifically the primers and probes from my RPA assay with slight modifications.
Anyone else? All right, thank you. Sonja Giardina is a junior at Hilo High, and her work is on coral bleaching.
Hi everybody, my name is Sonja and I'm from Hilo High School, as we all are, as you already know. I'm a junior and today I'm going to be presenting on the effect of fragmentation, isolation, and bleaching on coral reef fish assemblages, as you can see. Alright, so as many of you probably know, global warming has recently been leading to an increase in ocean temperatures, which has had many devastating effects around the globe, one of which being widespread coral bleaching, especially in Hawaii, where more than 50% of our corals have undergone bleaching events. So coral bleaching is affecting reefs across the planet, as I've mentioned, and it occurs when corals become stressed and they then expel their zooxanthellae. Zooxanthellae are symbiotic algae which provide them their nutrients and the energy that they need to survive. So if zooxanthellae are not able to recolonize the coral quickly, the coral then becomes bleached and dead. And as some of you maybe have been to the beach, you've seen the white corals versus the colorful corals. Those bleached corals are no longer alive. So although we do understand reasonably well the mechanisms of coral bleaching, we understand what happens, the zooxanthellae is expelled, there's less known about how fish are actually utilizing bleached reefs. And even less is known about how habitat fragmentation of these bleached reefs affects fish utilization. So what I was looking at in this project is to understand how fish are actually utilizing these previously living reefs, and specifically how fragmentation affects their ecological value. So specifically, I was looking at the size and distance from the main reef of variously sized coral fragments, and looking at how they compare to various characteristics of fish assemblages and fish utilization. So my problem question was, how does habitat fragmentation affect fish utilization of bleached reefs, as I have previously mentioned? So the various measurements that I looked at were abundance, species richness, evenness and diversity, and family richness, which I will go into later on in my presentation. So a quick overview of the site, since the term coral island that I'm saying is a little bit abstract. So here are some examples of the various coral islands that I looked at my survey site, which was Wailea Bay Marine Life Conservation District. This site was selected because it was a model study system. It already had, so here in these blue circles, these are what I was describing as the main reef area, so larger expanses of reef, whereas each of these plots is a coral island that I looked at. And as you can see, they are characterized by their distance from the main reef area and also their size. So here's a basic overview of the site that I was looking at and some examples to visualize the islands. So what I hypothesized that the bleached coral islands would still continue to hold ecological value to fish even though they were no longer living, because they would be important sources of shelter and habitat for the fish, not just specifically food, since they were no longer living. So corallivores would not necessarily be able to utilize them for food. I also hypothesized that larger islands would have a greater abundance and species diversity because they provide a larger and more complex habitat and therefore are able to support larger and more complex fish communities. Lastly, I hypothesized that islands closer to the main reef areas would also have a greater abundance and diversity of fish species because they are closer to the principal habitat. So, to test this experiment, I first constructed this mount here. You get to see me very large because the screen is huge. So in the red area, you can see the camera indicated. So basically what I did is I placed this camera mount next to each island. I started the video, I walked away, I swam away from the island and I allowed the video to record for 5 minutes. 11 islands were videoed for 5 minutes on 2 different occasions. So the 2 different occasions, it was different weather conditions, and I also changed up the order so I would be getting more variation in the condition of when I was filming. So after I did this, I went back and analyzed all the videos, I identified fish to the species, and then counted the number of individuals So how I did this was I found basically the maximum number of individuals of each species in a single frame of the video to kind of get a picture of maximum diversity and maximum abundance. And so the different factors I looked at, as I mentioned before, were abundance, species richness, evenness and diversity, and family richness. So abundance is basically the total number of individuals at each island. It doesn't take into account different species or anything like that, just a total population count. Species richness is how many species there were present at each island. Evenness was basically a measure of how similar counts between species were. So let's say we have a certain species that had 30 individuals present versus 5 individuals of another species. There was a bit of disparity in evenness, so evenness levels increased. Whereas if you have maybe 4 species in an island with 5 individuals across the board, that would be a 0 evenness level. And then lastly, for these species measurements, I calculated diversity as a function of the Shannon-Weiner Diversity Index, which is basically a mathematical formula that takes into account both species richness and evenness when coming up with an index that you can then use to compare populations. And lastly, I calculated family richness as well, which was simply the number of families. It is similar to species richness, but it does provide a picture of functional diversity as the species within one family are representing a certain niche in the population, and then showing how these islands are being utilized by various kinds of fish. And nonlinear regressions for statistics were used to analyze relationships. All right, there's a fun picture of me swimming around as we go into results. All right, so for abundance, which I mentioned before, is simply the total number of individuals. I found that it increases positively with volume. So I'll just go quickly over this graph. So here we have the volume of the island, which was measured using length and height measurements, and abundance, which is the number of individuals. And this graph shows volume increasing, and the islands are also classified as far, middle, and near distances from the reef. So I found that abundance does increase positively with volume, suggesting that larger islands are attracting greater number of fish. Secondly, I found that as you are moving further away from the reef, the abundance tends to decline with distance. One specifically interesting relationship I found in this relationship was that island 10, which doesn't mean much to you, but it is the island indicated in green, right there. So as I was going through, I was estimating coral cover of each island. And most of the islands tended to range from about 0 to 8%, so not a lot of live coral cover. Whereas this island actually had about 30% live coral cover as when it was estimated. Which was interesting because when I looked at the data later, I saw relationships, potential relationships, not enough data yet to confirm, but it did have higher coral cover, and it also tended to have almost 3 times more abundance than islands of the same size, which was interesting. And as you can see here, when I did the statistical analysis without the island 10, without the island with large coral cover, the relationship was actually more significant. So potentially that island is acting as an outlier, and the higher coral cover has affected the way fish utilize the island. So next I looked at species richness, which is the amount of species at each island. Species richness tended to increase with volume, suggesting that larger islands are attracting a greater variety of fish species. However, I also found that islands that were close to the main reef tended to have high richness regardless of their size. So here we move into the distance. Species richness did decrease with distance, suggesting that fewer variety of species are actually visiting the isolated habitat, so they're not traveling as far away from the main reef. Next, I looked at species evenness. So evenness did decrease with volume, but showed no significant relationship with distance, which is why it's not up here. So this can be explained by the fact that larger schools of fish do tend to aggregate at larger islands, which results in the disparity between counts of species. So at larger islands, I found schools with 60 or more fish, where at smaller islands, there were maybe a school of 18 or 15 fish. So with that 60 count of fish, it is going to result in more disparity. And that's why it tends to decrease with volume. So I also found that diversity decreased with distance. So as you move further from the main reef area, diversity of the coral islands are decreasing. But the main interesting thing that I found with this relationship was that the large islands tended to have comparable levels of diversity regardless of their isolation. So as you can see here, the two islands indicated by the squares, which was the large, they did tend to have similar levels of diversity even at different distances. So potentially, the larger islands, for what they're lacking in their proximity to the main reef, they're compensating for with their larger habitat resources and etc. They're able to provide a more complex set of resources for fish, and therefore fish are, let's say, willing to go travel that farther distance to utilize those islands. And lastly, I looked at family richness, which had relatively similar relationships as species richness. It increased with volume, and it was more of an index of functional diversity, showing that functional groups also tend to increase as islands get larger, and they also tend to decrease as you move further away from the reef. So in conclusion, the bleached reefs did retain ecological value. The death of the reefs did not necessarily decrease the fish utilization of the reefs, because there were fish present at each of the islands regardless of their coral cover. And I also concluded that the volume and distance from the main reef areas did significantly affect abundance, richness, evenness, and diversity. So there were relationships between these two factors and the various coral assemblage characteristics that visited these islands. So some applications of this research include insights about the conservation biology debate about single large versus several small. So in conservation biology, there's a debate both for terrestrial and for marine ecosystems, and restoring marine ecosystems and terrestrial as well, and creating conservation areas, of whether a single large area or several smaller areas are more effective at promoting diversity. So in this case, I would suggest that based on my data, I would suggest that restoring fewer larger areas of habitat instead of many smaller areas would be more effective at supporting fish diversity, due to the fact that the larger areas, even in isolation, were able to support high levels of complexity of fish communities. This data is also important to understand how fish are utilizing their habitat to inform potential restoration efforts, such as replanting coral from the lab or planting heat-resistant settlements, how to size and space these plantings. Also, insight into effective coral reef fragment size and spacing, which eventually, hopefully, will lead to conservation insight of how eventually what kind of habitat we want to prioritize when conserving to most effectively promote the diversity of fish communities. Whew. Thank you. I don't have an acknowledgement slide, but thank you, Ms. D, for helping during this process, and also Ms. Pinner. And any questions? Ina.
So, are you speaking to this side? You have a flight that's big?
Oh yeah, so it was, basically the evenness was 0 because there was only one species present. So it was mathematically impossible to get an evenness value just because there was only one species there, so. Yeah, it was excluded from the statistical analysis. Any more questions? Yes.
To better understand how coral bleaching affects this coral fish population, it would be better to compare this reef where there was, say, how much percent coral bleaching on the island?
The highest was 30, but all the others range from 0 to 8% approximately.
Compare like these reef islands with other reef islands with higher coral bleaching or less coral bleaching, or do a study where you like return to the same reef several years later to see how much.
Yeah, that's definitely a potential future direction for the study. In this location, there are other islands present that are further off the coast that do have higher levels of coral cover. So that would be an interesting future direction to take, to actually compare coral cover and fish utilization, which is definitely another relationship that would be interesting to analyze in the future.
Thank you, Sonja. Next up is Mya Lee. Mya is a freshman at Hilo High, and she is looking at SPF, the sunscreen, and antioxidant capacity of different substances.
Hi, I'm Mya Lee. I go to Hilo High School as everyone else here, but I'm the only freshman, so I'm like the baby, I guess. I investigated the SPF and antioxidant properties of spirulina, astaxanthin, fresh and powdered turmeric, and ginger. So these are all natural ingredients found here on this island, and I decided that it would be interesting to look into their SPF and antioxidant abilities. So the purpose of my project was to determine the SPF and antioxidant capacity of these 4 ingredients. I also wanted to see which natural ingredient would be the most effective as a natural sunscreen alternative, because natural products right now are really popular in the community. My hypothesis was that astaxanthin would produce the highest SPF of the 4 ingredients and also have the largest antioxidant capacity. So I did some research on each of the ingredients that I tested, and all of them kind of had similar talking about cancer and preventing skin cancer and that kind of stuff. So each of the chemical structures are shown at the bottom. I also looked into UV radiation and skin cancer because UV radiation and the sun hitting your skin is one of the main causes of skin cancer, and I wanted to look into these products to see if there is a way to prevent it with these natural ingredients. So I extracted samples from each of these ingredients and equalized them so they were all the same and they could be tested equally. I then went and used a spectrophotometer, which measures light absorbances, and I took the data collected from this machine and I plugged it into the SPF formula. From there, I went and completed a DPPH assay, which finds the antioxidant capacity. These samples were compared to the standard curve, which was made with gallic acid and DPPH. So I found that the SPF was, the ingredient with the highest SPF was the powdered turmeric and ginger at 7.5. And for a natural ingredient to have an SPF higher than 2, it is a good alternative. Fresh turmeric and ginger had an SPF of 0.9, astaxanthin with the SPF of 1.39, and then spirulina 1.59. For the DPPH assay, I had 3 plates, and the test was actually run 7 times because the standard curve was not matching with the samples, so I had to get the samples within the standard curve. I found that the powdered turmeric and ginger also had the highest the highest antioxidant capacity, followed by fresh turmeric and ginger, astaxanthin, then spirulina with a negative antioxidant capacity. And this kind of means that there are too many parts to spirulina because it's more of a complex ingredient. So the DPPH assay focuses mainly on more concentrated samples. This is plate 2, and the results all kind of showed the same thing with the negative spirulina, astaxanthin, fresh turmeric and ginger, and powdered turmeric and ginger. This is plate 3, and you can see the same kind of trends. This is a graph of the antioxidant capacity. This is the averages. So again, powdered turmeric and ginger was the highest. The conclusions were that the powdered turmeric and ginger had the highest SPF of 7.5, and it also had the highest antioxidant capacity of 77.39. And this shows that the powdered turmeric and ginger is a good alternative natural ingredient for sunscreen. The negative results for the spirulina doesn't mean that the spirulina is not a good antioxidant. It just means that a different type of assay could be used to change this. And the reason for the powdered turmeric and ginger and the fresh turmeric and ginger, such a large difference, is because the powdered turmeric and ginger was more fine, making it easier to get extracts from, versus the fresh was a little bit more chunky, and that made it a little bit harder. And for future research, I would like to conduct a polyphenol assay, which will determine what is happening with the polyphenol rings in each of the 4 samples. I would also like to test the strongest and most, the strongest ingredient and use that on melanoma skin cells. cancer cells, sorry, to see if it can protect skin cells from cancer. And that is it. Thank you. Any questions? Yes.
Are there any other natural ingredients that you're interested in?
So the turmeric and ginger were recommended by a friend, a family friend, and the spirulina and astaxanthin, my dad had been taking them, so I wanted to see if they actually had any antioxidants, because in the bottles, on the bottles, they have made claims that they protect your skin from the sun, and they have very high antioxidant capacities. Oh, yes.
So in your conclusion, you mentioned that part of the reason why spirulina might have shown is not effective because there might be other assays that work. Is there a specific reason why you chose this assay, or are there other assays in particular that you're looking for in the future that might show spirulina is higher, or potentially even your highest tolerance to
I chose this assay because it worked with most of my ingredients, but we did not know that the spirulina was so complex, so it wouldn't have the correct effect with the assay.
Yes, I have one. Would it matter if because you use methanol to extract? Is that
I use DMSO.
DMSO?
Yeah.
Would it make a difference if you used a different media to extract, do you think, or does that?
The DMSO works with the natural ingredients better, so I think that using a different kind of dilute would have had an effect on my results. Yeah.
You put turmeric and ginger together. Have you had any thoughts of turmeric?
No, because prior research has shown that together they have a stronger SPF. Yeah. Oh, okay. So the polyphenol rings would add to and show that they are a better natural ingredient to be used in a sunscreen. So it kind of promotes that. To show that there's more research behind it, it's good to conduct a polyphenol assay.
Thank you, Maya.
Thank you.
Last up this evening is Ina Klausner, and Ina also going to be talking about rat lungworm.
My name is Ina Klausner, and just like Elizabeth, I was working with the rat lungworm nematode. I'll breeze through the background research a little bit, but essentially it's the same as what she was saying. It's a parasitic nematode that can cause inflammation of the brain upon accidental human infection known as rat lungworm disease. I set upon my research to develop a death assay. Previously, there had been no specific way to determine whether or not a larva was live or dead effectively. So I was looking at specific fluorescent staining variables and optimizing them to form a most effective death assay. And then once I formed the successful death assay, look at preventative measures such as a vegetable wash to hopefully reduce the chance of humans becoming infected with the disease. So I was looking at the fluorescent molecule propidium iodide to detect Androstrongylis cantonensis death. And then upon optimization of that death assay with this fluorescent molecule, looking at hydrogen peroxide, apple cider vinegar, and vinegar to see if they could potentially effectively kill Androstrongylis cantonensis nematodes. These three solutions were chosen in particular because they are relatively affordable and easy for general members of the public to access, so that if successful, the public would be able to wash their vegetables and treat stuff accordingly. Okay. So this is the typical life cycle of a rat lungworm nematode. Typically, it only resides in rats and an intermediate host such as a slug. Human infection is most commonly occurred through ingestion of a slug or through anywhere that a slug has made contact with, and the rat lungworm has been transferred onto another thing. So as I mentioned previously, infection most commonly occurs through leafy greens. However, it can also occur through freshwater prawns, crayfish frogs, and even into people's catchment systems. So there are water concerns related to this as well. The unfortunate part of this disease is that there's no human-safe prevention mechanism known currently that we can effectively recommend to general members of the public to treat their vegetables with, and to know that it will make sure that it's safe for consumption. In addition, there's no known effective treatment for the specific disease that rat lungworm causes. There's only ways of treating the symptoms of the disease. Okay, so this is a brief description of the 3 different solutions that I was looking at and their chemical structures. Yeah, I'll move on. Okay, so I collected infected P. martensi semi-slugs from East Hawaii and isolated the rat lungworm larvae from these hosts through a water digest method, which is essentially just drowning the slugs. And then over the period of several days in this hypoxic environment, the larvae will settle to the bottom of a test tube, and then using a pipette, one can extract the larvae and view them under a microscope. So I was looking at 2 control and 3 experimental groups, a live and a dead control. Live being straight from the pipette into distilled water, dead being exposed to methanol and put into a negative 80 degrees Celsius freezer for 30 minutes. And then the 3 experimental groups were the 3 solutions I was looking at. Okay. So each of the solutions were added to 3 tubes. So I had 3 different trials for each. And then once I exposed the larvae to the solutions, I would remove them and then add the solutions again to ensure that all larvae were successfully exposed to the solutions. And the exposure occurred for 1 hour. After the solutions were removed, I added distilled water to each of the test tubes. Now, propydium iodide, the fluorescent molecule I was looking at, in previous years' research, I conducted this over the period of 2 years. I found that it was effective in determining Andriostrongylus cantonensis larvae death upon exposure to in 15 minutes. And then after exposing the larvae to the propydium iodide, I would view the effect of the staining in this high content image processing system known as the operetta, which essentially just excites the fluorescent molecules and then takes a picture of the fluorescence. So I would be able to get different images of the larvae exposed to solution. So I'd have a bright field image, as one would be able to see in a normal microscope, and to see the effect of the fluorescence. So this is a dead control exposed to propydium iodide. And as you can see, these are the fluorescing larvae. Okay. So this is the apple cider vinegar. As you can see, there are nematodes in the tube, but when you're looking just at the fluorescence, only the background material fluoresced. The same essentially occurred for the vinegar. There were larvae, but none fluoresced. And for the hydrogen peroxide, there did appear to be minimal fluorescence. However, that was not significant. So I then went on to quantify the fluorescence through the image processing system ImageJ. So I would take an image with yellow fluorescence and take it as an RGB image, only look at a specific channel, then select within a specific pixel intensity range. So I would be able to quantify the amount of pixels within the image I had fluoresced. And this method allowed me to only select for larvae and not any other background material that may have been in the well. So as you can see, there was very minimal percentage of larvae that fluoresced, except for the dead control, which indicates that the 3 solutions that I was looking at are not effective in killing rat lungworm larvae. And then when you look at this, there was some hydrogen peroxide exposed larvae that fluoresced. However, when you compare this to the dead control, it's simply not significant enough, especially considered that in a vegetable wash, people probably wouldn't be putting such a high concentration of hydrogen peroxide on their vegetables for such a prolonged period of time, because I exposed the larvae to them for a full hour. So although this experiment did validate the Propagation iodide death assay, vinegar, apple cider vinegar, and hydrogen peroxide do not effectively kill Androstrongylus cantonensis larvae. Although hydrogen peroxide may negatively affect nematode viability, it's not strong enough to be used as an effective vegetable wash. However, there are other promising solutions which can be used through the Propagation iodide death assay or potentially other death assays. So this could be other vegetable washes, or it could be looking at different treatment options such as antiparasitic drugs. And these are my acknowledgements. Thank you to Ms. D, Ms. Pinner, Dr. Jarvie, and Dr. Engel.
Questions for Ina? So Ina, your previous slide had the other potential applications. Do those work, or are those things that might work?
They could potentially work. In preliminary trials previously, the EpiClean showed promise. However, further investigation is necessary. In addition, that's a commercial vegetable wash, so that's it's recommended not for human consumption, not for a regular public use. So it'd probably be more at the level of a grocery store or a restaurant using it. The Department of Health does recommend washing your vegetables in water. It is good to put them under running water and to thoroughly wash your vegetables and to cook and freeze if possible. However, we don't have any solid recommendations we can make at this time. Thank you.
How long are those larvae outside?
They can live for several days. It depends on their environment and if you are feeding them additional material, because they will live off of the rat's brain tissue. So if they were being fed some kind of tissue or something to keep them alive, they could last in a petri dish for an extremely long time. However, if you're just leaving them alone and not feeding them anything, then they'll only survive for several days. However, that's still long enough that it could potentially cause some severe damage.
Thinking about
Mm-hmm.
Any other questions? Thank you, Ina.
I want to thank everybody who came tonight, especially Miss D. Miss D is a science teacher at Hilo High and hiding in the back there. Organized this class. All the parents for promoting this, and the mentors, Dr. Jarvie, who there's a few of the mentors in the room too. So thank you, everybody. But most of all, the 6 of you that had the courage to come up here and talk tonight, some of you for the 2nd year in a row for doing this. Thank you very much. We appreciate it.